Yes. Enzymes classification, transformation of enzymes, the class of enzymes. These are things we will discuss about the enzymes. These are the enzymes can be classified using numbering system defined by the enzyme commission. Okay. Here, according to the these systems consist of four-digit number, which classified based on the type of reaction enzymology. See, these are the IUBN. First one, enzyme, main important of the enzymes, borderly categorizing. One can categorizes enzymes into groups: hydrolyzing enzymes, desmolizing enzymes. Hydrolyzing enzymes, they catalyze the hydrolysis of substrate molecule. One second, first, the catalyze the enzymes is the substrate molecule. These enzymes are further subdivided into four types. First one is the amidase. Second one is the carbohydrates. Third one is the esterases. Fourth one is the proteases. Amidases, they hydrolyze amide and include asparaginase and urinase. The former enzymes hydrolyzes asparagine into ammonia and aspartic acid. The later enzyme hydrolyzes urea into ammonia and carbon dioxide. This is the amidase. Carbohydrates, they hydrolyze carbohydrates and include amylase, lactase, maltase, and sucrase. Amylase is the diesters, hydrolyze starch into maltose, lactose, hydrolyzes lactase into glucose and galactose. Maltose hydrolyze maltose into 2-glucose and sucrose hydrolyze sucrose into glucose and fructose. Third one is the esters. They hydrolyze substrate containing ester linkages and include chlorophyllase, lipase, nuclease, and phosphatase, chlorophyllase, hydrolyzes chlorophyll into chlorophyllide and phytol. Lipases hydrolyze lipids into fatty acids and glycerol phosphatase. Nuclease hydrolyze fructose biphosphate into fructose monophosphate. Phosphoric acid nuclease hydrolyzes nucleic acid into nucleotides. Next one, proteases. They hydrolyze proteins and include peptidase, protease. Peptidase hydrolyzes peptide into amino acid, and protease hydrolyzes protein into peptide. Second one is these are the hydrolyzing. Second one, desmolizing enzymes. They catalyze the reaction variously by removing of transforming group of atoms to substrate, from substrate and form one substrate to another substrate respectively. An account of chemical reaction, they catalyzed demonstrating desmolizing enzymes are following types. One is the carbolase, dehydrogenase, desmolase, epidermis, like this. Carboxylase, they catalyze the reaction by addition or removal of carbon dioxide. Carboxylase enzyme, they catalyze the reaction by addition or removal of carbon dioxide. Example, pyruvic decarboxylase and RUBP carboxylase enzymes. Pyruvic acid plus acetylide plus CO2, pyruvic decarboxylase enzyme, RUBP plus CO2, RUBP carboxylase enzyme to form the phosphoglyceric acid. Example. Second one is the dehydrogenase. They catalyze the reduction by transfer of a pair of H atoms. Examples are alcohol dehydrogenase and succinic dehydrogenase is a dehydrogenase enzyme. Acetaldehyde plus NADH, ethyl alcohol, NAD, succinic acid plus FAD, fumaric acid plus FADH2. Third one is the desmolases. They catalyze the reaction by breaking or lengthening of carbon atoms. For example, phosphoglyceroldehyde, dihydroxyacetone. This is the example for the desmolase. Epimerase, they catalyze the reaction by the convention of sugar it to epimer. Example of epimers, phosphofentose epimerase, which catalyze xylose 5-phosphate to ribulose 5-phosphate. These are the epimerase example. Hydrase, they catalyze the reaction either by addition or removal of water. Hydrase is the catalyze the reaction either by the addition or removal of the water. Common example are the enolase and fumarase. Enolase catalyzes fumaric acid into malic acid and fumarase 2-PG and PEP. Isomerases. Isomerases, they catalyze the interconvention of aldolase, ketose, sugars. Common example isomerase, phosphoexoisomerase, phosphotriose isomerase. For farmer catalyze glucose 6-phosphatase, fructose 6-phosphatase, later phosphoglyceroldehydes, phosphordihydroxyacetone. This is the isomerase enzyme example. Oxidase, they catalyze oxido reductions using a molecular oxygen as an acceptor. That is, they remove electrons from substrate molecule and give them a molecular oxygen. Example is cytochrome oxidase. Next one, peroxidase. They catalyze the reaction by acting on hydrogen peroxide or organic peroxidase only in the presence of a substrate that the accept of oxygen. Example, all catalysis enzymes. Next one is the transmination. They catalyze the reaction by the transfer of amino group from one molecule to another molecule. For common example, alanine amino transferase, which catalyze the conversion of alanine plus ketoglutanic acid into pyruvic acid and glutamic acid. This is a transminase. Transphosphorylase. They catalyze the reaction by the transfer of phosphate group from one molecule to another. For example, hexokinase catalyzes reaction of glucose AT plus ATP into glucose ATP into glucose 6-phosphate plus ADP, and pyruvic kinase catalyze reaction of PEP ADP into pyruvic acid plus ATP. These are the example for all. But what is the classification? Different classes of enzymes. IUB system enzyme classify according to International Union of Biochemistry, IUB, International Union of Biochemistry, system of classification based on their reaction specificity. Enzymes are grouped in 6 major classes. First one, oxidoreductase, transfer of H atoms, transfer of H atoms of electrons. H atoms meaning transfer of electrons, redoxon reaction. Transfer of protons or atoms of electron. In this class are included those enzymes that catalyze both biological oxidation and reduction. Common example are catalyzes, dehydrogenase, peroxidase, oxidase. These are the oxidoreductase. Second one, transferase. Transfer of small group between molecules. Transferase is a transfer of small groups between molecules. In this class are include those enzymes which catalyze the transfer of different groups from one molecule to another molecule. Common example are amino acid, amino transfer, phosphotransferase enzyme. Phosphotransferase enzyme. This is a transferase. Next one is that one hydrolyze enzyme. In this one, hydrolyze enzyme hydrolyzes hydrolyzes of the substance, hydrolyzes of the substrate molecule. Common example include are amidase, carbonase, esterase, proteases. These are the hydrolysis enzyme. Mhm. Next one, lyases. Lyases enzyme lyase enzyme additional or removal or to additional or removal to form double bond. In this class are include in this lyases in this class removed in this class are include those enzymes that catalyze respectively addition or removal of groups of atom to double bond from double bond form to double bond. Examples these lyases carboxylase, desmolase, hydrase. Lyases example. Next one is the fifth one isomerases. In this class are included those enzymes that catalyze that intermolecular reagent. Common examples for isomerases incorporated into this class are epimerase, isomerase, intramolecular transferases. This is called the isomerase. Next sixth one is the ligases. Synthetase. Another name ligases is the common name, and another name is called synthetases. Formation of bonds between C and other atom using ATP as an energy source. In this class are included those enzymes that catalyze the linking of two compounds coupled to breakdown of pyrophosphate bond in ATP or similar compound. Common example of common example are acetyl-CoA carboxylase, amino acyl synthetase. Example for the synthetases are ligases. These are the IUB classification. India International Union of Biochemistry classified the enzymes 6 types: oxidoreductase, transferase, hydrolases, lyases, isomerases, ligases, or synthetases. Okay. Another thing is the enzyme common number. What is the EC? EC meaning enzyme common number. Commission, this is the enzyme commission. Commission number, not common, commission number. All the enzymes are given systematic name and number which identifies them in technical literature. They may also have a common name such as trypsin or renin. Okay, these names are given the enzyme commission. That is the enzyme commission. The name and the number are systematic description of what the description of what the enzyme does. The number is a 4 digit. The number is a see. Then 1 second. The number is a 4 digit. The number is a 4 digit number. The first number description of this one is the first number description of class. The first number is the description of class. This is the class. This is the class or description of class out of 6 major classes. Already above mentioned, no? The second number already above I'll mention 6 classes. The second number Describe the this one. The second number describe the class. Describe the subclass. Class, subclass. The third number describe the subclass, subclasses. And last, fourth number describe the reaction catalyze. Enzyme number in the subclass. This is the, for example, I'll explain to the this one. Next one, coming to the another one here. The example for reaction, for the example, I'll write here 2, 7, 3, 2. Okay, here example is there, but two is there. No, this two, because a transfer group from ATP creatine, ATP creatine, these two. The seven become, because the group phosphate 2.73 means this 2.73 means the subclass which transfer to the phosphate to nitrogen atom. Here the dots are essential since some classes of enzymes have more than 10 number. The systematic name is ATP creation phosphate transfer, the enzyme which transfer a phosphate group from the ATP 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 creatines. Next one, some example, oxidoreductase. Acting oxidoreductase is the, for example, 1,1H. What is the oxidore? 1, 1, 1. Hydrogen acceptor NADP. Alcohol acceptor NADP. Alcohol lactic acid like this. Another one 1.2.1. This is the another example. This is the oxidation. Transpiration 2.2.1. 2.6.1. 2.7.1 is the second. Hydrolyze third one. 3.16. 3.4. Lyases 4.1, 4.12 like this, carbonitrase. And isomerase is the fifth class, no? 5.11, 5.13. Last one is the lyases, 6.2, 6.2.1 like this, all the lyases. Okay, genetic engineering requires certain enzyme which help cutting the modifying joining of the DNA. The DST Nicole divide the enzyme into three classes. Generally, this is the class. This is the class, subclass, subclass. This is the indicate specific enzyme name. Coming to the next. What is the oxidoreductase? See, oxidoreductase acting oxidoreductase acting on CHOH group of donor. CHOH group of cis one is the CHOH group of donor. CH which donor? CH and OH. CHOH. CHOH group of donor. Here, hydrogen acceptor is a NADP, NAD plus NADP. Hydrogen acceptor, which one? NAD plus. NAD plus to NADP. NADP plus, NADH plus to NADP plus. Hydrogen acceptor. NAD oxidoreductase, NAD oxidoreductase malic dehydrogenase, NAD oxidoreductase lactose dehydrogenase, NAD oxidoreductase alcohol dehydrogenase. Acting as 1,1,1,1. Alcohol dehydrogenase is the best example. This one, 111. And next one is the cytochrome oxidases. Cytochrome oxidases acting alpha keto or keto group donors, glyceraldehyde phosphate and amide acid oxidoreductase. Next one, CHCH group donor with accept succinic acid, fentaric acid. These are the best example for the oxidoreductases. Second one is the transferases. Here, the transferases enzyme, the transferase enzyme, the best example, uh, transfer aldehyde or ketone residues. Transfer aldehyde or ketone residues. Example 2.7.11 or 2.2.221. 2 point exokinase, this one. Exokinase. In case here another examples are there or involve transfer from group between molecule. Between molecule means aldehyde to or ketones residues. Aldehyde to ketone. 2.2 one is the best example. Pseudoheptulose phosphate. This is a pseudoheptulose phosphate, glyceraldehyde phosphate, glycolaldehyde transferate. Next one, 2.2.12. 2.2.12 pseudoheptulose phosphate, glyceraldehyde phosphate dehydrogenase acetone transferase enzyme. Transfer nitrogenous group, that is the 2.61 aminotransferase, ketoglutaric aminotransferase, asparatic aminotransferase. Next one, 2.71. 2.7 alkyl group of serves as acceptor. Transfer photophosphorus containing group 2.7.1. Hydrolyze. Hydrolyze meaning the third one 3.1.6. That is called the hydrolyze enzyme. These hydrolyzes serving or ester bond 3.1.6 the phosphoric moisture hydrolyase enzyme 3.1.689 glucose 6 phosphate phosphohydrolysis. Next one is the 3.4.4. This is a serving on peptide bonds. Peptide, peptidohydroxylase. Pepsin, trypsin is that one. 3.5.1. Serving CN bonds and other peptide bonds. 3.5.1.5. This is the urea amido hydrolysis enzyme. Next, fourth one, 1 second, I'll love it. Fourth one is the lyases. Coming to the fourth one is the hydrolysis is completed. Fourth one is the lyases. Lyases is the carbon-carbon lyase. Carbon-carbon lyases is the lyases. For example, this one. For example, this one. 4.1.1 carboxylase 4.1.1.1 keto acid carbolyase. This is the pyruvic decarboxylase. 4.1.27 aldolase. This one is a aldolase. Aldolase keto 1 phosphate dehydrate hydrolase is called a aldolase. 4.2.1.2. 4.2.1.2 2.1.2, 4.2.1.2, malate hydrolase. This is the carbon oxygen lyase. Next one, carbon 4.3.1 is the aspartate, aspartate or aspartic acid. And histidase. And next one is the histidase. Coming to the last, next one, isomerase. Hmm. Racemose and epidermis. Racemose and epidermis. Racemose is the 5.1.1 serving on amino acid and derivatives. 5.1.1.1 alanine racemose serving on carbohydrate and derivatives. 5.1.3.1 ribulose 6 phosphate 3 epimerase enzyme is the isomerase. Coming to the lyases. Lyases forming cis and C bonds, CS bonds. CS bonds, acetic as the acid triol synthetic. 6.2.1.1 acetate coenzyme A synthetic acetyl co-synthetase forming CC bond 6.4.1.2 acetyl CoA carbon dioxide lyases acetyl CoA carboxylase. This is the lyases. Okay, these are all the classification of the enzymes. Now we will discuss about to the enzymes and biochemistry. What is the enzymes theory are there? How to enzymes involving theories? We will discuss about to the theories of enzymes. One second, I'll share my PPT. Total, now enzyme, what is the enzyme, structure of enzymes, all are the complete. Structure of the enzymes and what are the enzymes are completed. Classification of enzymes also now complete. Now we will discuss about to the, one second. Nomenclature of enzymes, isoenzymes, allosteroenzymes, multizymes complex, hydrolyzing enzymes, major classes. Major classes already complete the total major classes of the enzyme. And the properties of the enzymes, different enzymes properties, catalytic properties are there, the mode of enzyme action, catalytic catalytic of enzymes. One second. Properties lo first one, catalytic of enzymes are catalytic. Here, enzymes are very similar to inorganic catalysis. They are present in very small quantities and very small quantities. And these are they are present in very small, they are capable They are capable for the they are capable they are capable for capable of bringing about the change in a large amount of the substrate active site and 1 second, I'll share my PPT. See, these are things are already complete. These are the things are already complete. Enzymes, that is the catalytic activities, isomerases, enzyme active. Next one is the where the isosite discover the enzymes. These are things, enzymes classification mode of axons. Enzymes biocatalysis biochemical reaction in vivo and in vitro. They are highly specific substrate and great catalytic power. That is, enhance the rate of reaction tremendously without being changed. All the enzymes are proteins without expansion, some small group of catalytic. That is the RNA molecule called ribozymes. What are they? Ribozymes like proteins, molecular weight of the enzymes range from 2000 to more than 1 million daltons. Enzymatic activity of proteinous enzymes may be affected depending on the conformational structure, conformational structurals as well as denaturation. There are many enzymes which require cofactors for the catalytic activity. Cofactor may be complex organism molecule called coenzymes or metal ions such, for example, iron, magnesium, zinc, manganese, coenzymes. An enzyme plus its cofactor is called holoenzyme. In such cases, the protein component in cofactor required enzyme is called apoenzyme. Very, very important. The protein component in cofactor required enzyme is called the apoenzyme. Apoenzyme, see, this is the some cofactors, coenzyme, precursor, vitamins, and their role. First one, coenzyme, biosin. Biosin is a coenzyme. This is a coenzyme. The precursor is the vitamin B7. That is a biotin. Vitamin B7 meaning biosin. What is the role of action? Role of action is transfer of CO2. Transfer of CO2. The next one is the coenzyme B12. This is the vitamin B12 transfer of an alkyl group. The coenzyme flavin adenine dinucleotide, FAD, is called the coenzyme. That coenzyme is a flavin adenine dinucleotide enzyme. Flavin adenine dinucleotide enzyme. This precursor vitamin is a B2 riboflavin. What is the role, catalytic role? Transfer of the electrons, this coenzyme. Coenzyme A, pantothenic acid, vitamin B3, transfer or acyl and alkyl group. Next one, nicotinamide, NAD, nicotinamide diadenine dinucleotide. Nicotinamide diadenine dinucleotide. This is the vitamin C, niacin. Niacin, vitamin B5, transfer of the hydrates, H+. Next one, coenzyme, pyridoxal phosphate. This is the precursor, pyridoxine, vitamin B6, transfer of amino group. Next one, coenzyme, thiamine pyrophosphate. Thiamine B12, B1, transfer of aldehydes. Next one, coenzyme, tetrahydrofolate. This is the folic acid, vitamin B9, transfer of one carbon group. Coenzymes take part catalyze transiently and are carriers of specific functional group. Most of the coenzymes are derived from vitamins, organic nutrient. What is the metal ions? Iron, sulfur, Fe, metal ion, ferric acid, ferric, catalyze pyridoxine, cytochrome oxidase, cupric acid, cytochrome oxidase, magnesium, DNA polymerase, manganese, arginase, potassium pyruvic kinase, molybdenum nitrase, nitrose reductase, zinc carbonic anhydrase, alcohol dehydrogenase, nickel urease. When coenzyme or metal ions tightly bound through covalent bond with the enzyme protein is called, that is the prosthetic group. Now already classification is completed, yes or no? The International Union of Biochemistry, enzyme to transport 6. Next one is the isozymes. Many enzymes are present in the multiple forms. Many enzymes are present in multiple forms, more than one molecular form. In the same species, same tissues, or even in the same cell also. Many enzymes are present in multiple forms, more than one molecular form, in the same species, same tissues, or even in the same cell. These enzymes are called isoenzymes or isozymes. Isoenzymes catalyze the same reaction but have different amino acid compositions. Hence, this process different physiochemical properties. For example, glycotic enzyme hexokinase exists for four isomes formed in various tissues. Similarly, lactase dehydrogenase involved in anaerobic glucose metabolism has two isozyme forms in human. One is present in heart, other is present in skeletal muscles. Where is the enzyme active site? Enzyme active site, the catalytic reaction performed by enzymes occur at a particular site on the enzyme. This site name is called active site. What is the active site? Catalytic reaction performed by the enzymes. Catalytic reaction performed by the enzymes occurs at a one particular site on the enzyme. This is the active site and represent only small part of the total size of the enzyme. The active site is a clearly defined pocket or cleft in the enzyme molecule where the whole portion or substrate can fit. It active site as a three-dimensional structure since it constant of portion of a polypeptide chain. Various non-covalent bonds involved in enzyme substrate binding are electrostatic interactions. Hydrogen bonds, Van der Waal force hydrophobic interaction, the active site compressions with the non-polar environment. These facilitates binding the substrate side of the catalysis. However, some polar residues may be present. This type of environment is not found to the other region of the enzyme molecule. The first theory is a lock and key theory. Feature lock. This theory is also very important. Interaction between enzyme and its substrate according to the lock and key theory model. This lock and key theory model in 1894 introduced by lock and key theory model for Fisher scientist M.L. Fisher. M.L. Fisher scientist. This enzyme interaction was proposed by the scientist named M.L. Fisher. M.L. Fisher, according to this model, complementary structure features are present between enzyme and substratum. See, this is the enzyme. This one is the substratum and the active site is pre-shaped. where the active site is the pre-shaped to fit the substrate, and the active site is pre-shaped, this one to fit the substrate. The substrate can fit into the complementary site on the enzyme as a key fits into a lock. This result in the formation of enzyme substrate complex. This is the enzyme to fit the clearly. That's why this is the lock and key model. Next one is a Coastland induced fit model. Second one is a Coastland. Daniel Coastland, scientist, in 1958 proposed induced fit hypothesis. What is that one? Induced fit hypothesis. He suggested that the structure of a substrate may be complementary to that of the active site in the enzyme substrate complex, but not in the free enzyme. But not in the free enzyme. The interaction between the substrate and the enzyme induce conformational changes in the enzyme which elegance that amino acid reducing or other groups for substrate binding catalyzes or both. The relationship between a substrate and active site resemble hand and wooden glove. During interaction, the structure of one component mean that is substrate or hand remain rigid and the shape the second component that is the active site or glove flexible to become complementary to that of the first. What is the specificity? Enzyme specificity in the action. And all enzymes are highly specific in action. In fact, the properties that make enzymes such as a strong catalysis are their specificity of substrate binding and their ideal arrangement of catalytic group. Various type of enzyme specificity are grouped specificity, absolute specificity, stereospecificity, and geometrical specificity. When enzymes act on several different closely related substrate, then it is called group specificity. When enzymes act only on one particular substrate, it is called absolute specificity. Next one, stereochemicals or optical specificity occurs when substrate exists in two stereochemical forms, that is chemically in a identical but different arrangement of atoms in three-dimensional shapes. Then only one of the isomer will undergo to the reaction by particular enzyme. For example, D-amino acid oxidase catalyze the oxidation of the D-amino acid to keto acid. In geometrical specificity, enzymes are specific towards cis and trans forms. Cis form and transfer. For example, fumarase catalyzes the interconvention of fumarate and malate. What are the factors affecting for the enzyme activity? And what is the main here? What type of factors involving the enzyme activity? That is the physiological properties or other properties. The rate of enzyme catalyzed reaction is influenced by changing the environmental conditions. The important factors that influence the velocity of enzyme catalyzed reaction, that is temperature, pH, substrate concentration, and modulators. What is the temperature? How much of temperature is the optimum temperature? See, the rate of an enzyme catalyzed reaction increases with the increase in temperature up to maximum and then fall. When a graph is plotted between temperatures versus enzyme activity. See, this is the graph. This is a temperature versus reaction reaction velocity, temperature and reaction temperature versus enzyme activity. A bell-shaped curve is obtained. You can observe the what shape is obtained. One bell shape curved is obtained. The temperature at which the maximum rate of reaction occur, it is called enzyme optimum temperature. This is called the enzyme optimum temperature. This temperature which maximum rate reaction occur is called the optimum temperature differ from different enzymes. But most of the enzymes, it is between 40 degrees centigrade to 45 degrees centigrade. Majority of enzyme in human body have an optimum temperature of around 37 degrees centigrade. Okay, that is 98.6 foreign degrees. The human body optimum temperature is 37 degrees centigrade or 98.6 Fahrenheit and are denatured or degraded at extreme temperature. The enzyme is when the extreme temperature is there, denature or degrade of the temperature. However, the enzymes like TAQ DNA polymerase present in the thermophilic bacteria, Thermus aquaticus, venom phosphokinase, and muscle adenosine kinase are active even 100 degrees centigrade also. Which one is 100 degrees centigrade to bear the enzyme? TAQ DNA polymerase. Thermophilic bacteria. You know, high temperature are this, they lie also living position. And the next one, Thermus aquaticus. Next one, Venom phosphokinase and muscle adenylate kinase are active even at 100 degrees centigrade. C. Next one, hydrogen ion concentration. pH meaning, what is the pH? Hydrogen ion concentration. Enzyme activity is also affected. The second effect by the pH. Enzyme activity is also affected by pH. A plot of enzyme activity against pH result in a bell-shaped curve. What is the curve is there? Bell-shaped curve. Each enzyme as it is unique optimum pH at which the rate of reaction is greatest. Rate of reaction is greatest. The optimum pH is the pH at which the activity of a particular enzyme is at maximum. Many enzymes of higher organism show optimum reaction. So, optimum reaction rate around natural pH 6 to 8. However, there are several expansion such as pepsin, the enzyme, for example, several. One is a pepsin. The pH is only 1 to 2 acid. The phosphatase enzyme pH level only optimum pH 4 to 5. And other one alkaline phosphatase. The pH level 10 to 11 only. Okay, this is the for example pH. Below and above the optimum pH, the enzyme activity is much lower and extreme pH, the enzyme become totally inactive. Third one, substrate concentration. What is the next one? Substrate. The substrate concentration also influence the enzyme activity. As the substrate concentration increase, the rate of reaction also increase. This is because the more substrate molecule will interact with the enzyme molecule. That's where the more products will be formed. However, after a certain concentration, further increase in substrate concentration will have no effect on the rate of reaction. Since the substrate concentration will no longer be the limiting factor. At this stage, enzyme molecule become saturated and work at their maximum possible rate also. This is the these all are the factors, the enzyme activity factors, temperature, pH, and substrate concentration. Now, unit of enzyme activity. What is the unit of enzyme activity? Unit of enzyme. The enzyme unit is the amount of enzyme that catalyzes the conversion of 1 micromole of substrate per minute under stranded conditions. The International Union of Biochemistry, IUB, adopted enzyme unit as unit of enzyme activity in 1964, but it was discouraged in favor of the Katal. Since favor of the Katal. Since this is the favor of the Katal. Since the minute, here the minute is not an SI unit, that is called the SI unit. One katal meaning cat is the amount of enzyme that catalyzes is the 1 mole of substrate per second. So 1 pot is equal to 60 billions units are present in this one. Specific activity. Specific activity is the another specific activity is the another common unit of enzyme is specific activity. It is defined as the moles of products of products formed by an enzyme in a given amount of a time. Here time is the minute. Time is the minute under given conditions per milligrams of proteins. Specific activity represented a measurement of enzyme purity in the mixture. Coming to the next one is the mechanism of enzyme action. One, for understanding the enzyme mechanism, you should consider two thermodynamic properties of a reaction. These are free energy difference between the products and reactant and the energy needed to initiate the conversion of reactant into product. The former energy, that is delta G, determines whether the reaction is spontaneous or whereas the later determine the rate of reaction. The enzymes affect the energy which determine the rate of reaction. The enzymes cannot change law of thermodynamics and therefore cannot alter the equilibrium of a biochemical reaction. They speed up the attainment of equilibrium. Rate of reaction rather depend on the free energy. Free energy and free energy of activator, that is delta GA, that is called the delta GA, which is not related to delta G. The substrate S of a reaction converted into product, that is called the P, converted into product is called the P formation of a formation of a transition state has higher free energy than either S or P, either SRP, the difference between the free energy than either SRP between the free energy of transmission state and substrate is called, this is the Gibbs free energy of activation or simply activation energy, that is delta GA. The enzyme enhance reaction rate without alternating alternating delta G of reaction. Rather, they lower the activation of delta GA. That is the next one is the kinetic kinetics of energy, kinetics of enzyme catalyzed reactions. Kinetics of the enzyme catalytic reactions. During catalysis substrate. During catalysis substrate, S bind the active active site of the enzyme E and result and result in the formation of a enzyme substrate complex, that is the ES, which is finally converted into product, that is called P. The reaction can be this reaction can be represented as the formula E plus S equilibrium. E plus S is the equilibrium. ES plus E plus P. You know what is E? What is the P? Enzyme E, P is the product. When E forms, when E forms weakly bonded complex, that is ES. That is called the ES with the substrate S. The ES complex composed to yield product P. To yield product P, the free enzyme E. The kinetic enzyme catalyzed by reaction was explained by the scientist Lenore Mitchell and Mount Menchan in 1913. The most remarkable features of this kinetin is that specific ES complex, ES complex is an this one is an intermediate during catalysis. Mitchell-Mentor theory of enzyme kinetic is the simplest one that accounts for kinetic properties of many enzymes. Further suffering Further simplifying the reaction, further simplifying the reaction, Mitchell and Mentor derived following equations for one substrate reaction. That is called V0 is equal to V maximum K maximum. The equation is called Mitchell mention equation, where Km is called Mitchell constant. V0 is initial velocity. V maximum velocity maximum, velocity of reaction, substrate concentration, substrate concentration. A graph of V0, that is the against, result in the rectangular hyperbola. This is the maximum. The graph we can see that very low substrate concentration. The reaction rate is the maximum independent. The which of the maximum reaction rate is obtained? The maximum velocity represented the turn of that one, the turn of name enzymes. This is the turnover number. This one is called the turnover number, is the number of substrate molecule converted into product by an enzyme molecule in a unit time. It is equal kinetic constant. This is the K2, which is called Kerlik. Next one, enzyme inhibitors. How many enzyme inhibitors are there? Substrate, substance substance which decreases the rate of enzyme catalyzed reaction are called enzyme inhibitors. What is the enzyme inhibitors? Substance which decreases, substance which decreases the rate of an enzyme catalyzed reactions are called enzyme inhibitor, and the process is known as enzyme inhibition. That is called the enzyme inhibition. Enzyme inhibited can be classified into two types. One is the reversible inhibition. The second one, irreversible inhibition. First one, in reversible inhibition, the reverse inhibitor bind very tightly to the enzyme and does not disassociate from it. For example, the antibiotic penicillin act as the inhibitor. And you can use the antibiotic, no? In that, antibiotic act as a inhibitor and bind with the enzyme transpiration. Bind into the inhibition enzyme inhibitor bind with the enzyme transpeptidase. That is the transpeptidase, which is transpeptidase enzyme, which is the responsible for synthesis of bacterial cell wall, which is the responsible for synthesis of the which sensible of the enzyme is a synthesis of the bacterial, synthesis of the bacterial, which is synthesis of the bacterial cell wall. Okay, thus killing the bacteria. That's why the killing the bacteria. Killing the bacteria. Next one, in the same way, the drug aspirin, second drug, the drug aspirin the drug aspirin inhibits the enzyme cyclooxygenase, cyclooxygenase, thus reducing the inflammation. In reversible inhibitors, reversible inhibitors radially, rapidly dissociate from the enzyme inhibitor complex. There are three types of reversible inhibitor. One is the competitive inhibitor, non-competitive inhibitor. Third one is the uncompetitive inhibitor. What is the competitive inhibitor? Competitive inhibitor, there is close resemble in the structure of inhibitor 1 and substrate S. Therefore, they both uh, compete for the same active site on the enzyme. The enzyme can form enzyme. The enzyme can form enzyme substrate, ES substrate, ES complex, or it can form enzyme inhibitor EI complex, but not both ESI. Competitive inhibitors, competitor inhibitors decrease the rate of reaction by reducing the amount of active enzyme molecule bond to a substrate at very high substrate concentration. The change binding inhibitor molecule to the enzyme will be reduced. So V is the maximum of the reaction. Maximum of the reaction will not be changed. However, the Km is which is substrate concentration at which Vx increased in presence of a inhibitor and denoted symbol that is called K3M. Okay, this is one. See, this one. Inhibitor. This is a competitive inhibitor. Second one is the non-competitive inhibitor. Non-competitive inhibitor is called the, in this type, inhibitor has no structural similar which substrate and bind with the enzyme at different site and other than the active site. Therefore, there is no competition between S and I, and formation of ES, EI and ESI takes place here. The inhibitor 1 and substrate S can bind simultaneously to the same enzyme molecule as their binding site are different and hence do not overlap. Non-competitive inhibitor lower the V maximum rather than by decreasing the proportion of enzyme molecule that are bound to the S. Thus, the non-competitive inhibitor, in contrast to competitive inhibitor, cannot be overcome by increasing substrate concentration. The substrate can still bind to the EI complex. However, the ESI does not from product. The I effectively lower the concentration of active enzyme and hence lower of the Vmax. There is no effect on Km as the inhibitor decrease the amount of functional enzyme. The third one, uncompetitive inhibitor. In this type of inhibition inhibitor does not bind the free enzyme. It binds only. See, this one is the enzyme substrater. E is the enzyme. S is the substrate. ES enzyme substrated. P is the enzyme plus product. Okay. This is a EI enzyme inhibitor, enzyme substrate inhibitor complex. This is a enzyme substrate, ES. Substrate enzyme, this is a inhibitor. Next one, enzyme inhibitor complex. These are all three are the non-competitive inhibitors. This is the inhibitor. Next one, allosteric enzymes. Allosteric enzyme do not obey Mitchell's mention kinetics. This is the not obey for that one. These enzymes in general consist of more than one protein subunits. Therefore, more than one active site are present. Allosteric enzyme result in the sigmoidal graph instead of rectangular hyperbola when B0 is plotted against substrate concentration S. Each subunit of allosteric enzyme also contain regulatory site along with active site. Regulatory molecules may reversible bind to the regulatory site and alter the affinity to enzyme for substrate binding, whether as most of the enzyme obtaining for the Mitchell mission kinetics. This is a normal enzyme, allosteric enzyme, the key regulators of metabolic pathway in the cell. See, enzyme substrate, enzyme and product. This is a ESI, enzyme substrate inhibitor. This is a enzyme, this one is a substrate, and this is a inhibitor. This total is called uncompetitive inhibitors. This is the kinetic of the allosteric enzyme, the substrate AO2. Next, brief introduction of the to bioenergetic. Biological energetic, bioenergetic deal with the transformation and use of energy by living cell in biological reactions. The energy is released as the reaction move from a higher to lower energy level. Energy is liberated in the form of heat. That is the A and X. That one is called the A and X. B, AB conversions of metabolites AB occurs with the release of energy. It is coupled to another reaction in which energy is required to convert metabolism. That is called the XY. Another thing is the this energy thermodynamics also present. What are the thermodynamics? Law of thermodynamics is the most important for the enzyme. Here, energies are thermodynamics and other conclusions are present in this one. Okay, here in this one, you can observe the energy is the capacity to work and exit in the variety to form such as the electrical energy, mechanical energy, chemical, heat energy, and light energy. These forms of energies are interconvertible. Bioenergetic is concerned with the changes in the energy during biochemical process. It does not deal with the mechanism or speed up of the process. This thermodynamics is the branch of physical chemistry that deals with the energy changes. There are two fundamental laws of thermodynamics. This explain to the interconversion of various forms of energy. The two laws help in understanding the direction of a reaction, whether forward or reverse. This is the first. The second one, accumulation of work, whether useful or non-useful. Third one, whether energy for executing a reaction must be delivered from an external sources. The first law of thermodynamics, according to this law, energy exchange in any process takes place between system and surroundings. System is a matter within the defined region and surroundings constitute matter in the rest of the universe. Thus, the system and surrounding make the universe, which includes the entire earth and outer space. The first law of thermodynamics states that energy can neither be created nor destroyed. In the first law, the energy can be created, neither can be, the energy can neither be created nor destroyed, but can be converted into other forms energy. It means the total amount of energy in the universe. This is the system plus surrounding remain constant. Energy E is equal to EB minus EA is equal to Q and W. E is the change in internal energy. E is called, whether the E is called change in internal energy. EB is equal to energy of a system at the end of the process. EA energy of a system at the start of a process. A is the start of process. What is the Q? Q is the heat absorbed. Heat absorbed by a system. Heat absorbed by a system is called Q. Another one is the W. W is work done. What is the W? W meaning work done, meaning that is the work done by the system. W is called the work done by the system is called the W. According to this equation changes in the energy of a system depends only on the initial and the final stage and not the path of transformation. The second law of thermodynamics, the first law of thermodynamics cannot be used to predict the spontaneous or non-spontaneous of the reaction. On the other hand, the second law introduced a term entropy, that is called the entropy, which is denoted by symbol S. Entropy is degree of the randomness or disorders of a system. This is the explain where the reaction takes place spontaneously. Entropy is the become a more disorders. Entropy reach maximum system, equilibrium, biological system. Another thing is do not follow the second law. Either the second law as the life state, higher organism, lower entropy rather than increase entropy. This higher order form of life maintained a while constitution chemicals energy. This is the second law of the energy. Combining two laws therefore a different thermodynamic function. Free energy is used to overcome this problem. Free energy is denoted by symbol G. This is a Gibbs. This is 1879. The Gibbs created free energy function by the combining the first and second law of thermodynamics. Following equation was obtained. G is equal to H is T. G for meaning G is the change in the free energy of a reaction system. H is the change in the heat content of enthalpy of this system. T is the absolute temperature at which the process is taking place. S changing the entropy of the system. This is the ATP universal currency of the energy. This is the total enzymes are the completed. Thank you.